Namaskar. Welcome back to the lecture series on sericulture. In this session, we are going to see the molecular biotechnology in sericulture. Sericulture in India is practiced mostly in the tropical regions involving 6 million people. It is labor intensive and also improves the socio-economic status of small and marginal farmers. However, several problems like disease occurrence, unhygienic conditions during rearing, low nutritive quality of leaf fed to the silkworms, usage of primitive silk reeling and weaving machineries have kept the sericulture in India economically unattractive. The silkworm breeding is a complex and challenging task as most of the quantitative traits are polygenic, which are closely related and associated. Hence, to develop an improved silkworm race with all these complex characters is extremely difficult and time-consuming task. However, the recent research and development efforts have enabled substantial improvement in the bivalve and silkworm quality, productivity as well as stability of the silk production in India. The silkworm is a convenient model for the studies of the molecular basis of development and gene expression using the tools of biotechnology, molecular biotechnology. The production of bivalve cocoons and silk in India is important to upgrade the quality and productivity of Indian silk to international grade, which is yet to become a reality. Hence, it is necessary to venture and utilize the recent advances in the molecular biology in addition to conventional approaches. The recent advances in the biotechnology like recombinant DNA techniques, genetic engineering through manipulation of desirable traits, stem cell work have revolutionized the research in plant and animal sciences. Genetic engineering leads to develop better varieties of silkworm that produce more silk and are more resistant to diseases and environmental fluctuations. Let us see the better races of the silkworm. The quality and yield of silk depend on availability and healthy silkworms, which itself depends high quality feed and disease-free stocks. With the spread of sericulture to different parts of the country, it had become necessary to adapt the silkworm to local rearing conditions. Traditionally, strain improvement has used the breeding methods for the production of new high-yielding strains with such desirable traits as disease resistance and heat tolerance. Where intense breeding techniques studies have been carried out for long periods of time, traditional approaches appear to be reaching the natural biological limits of the silkworm with regard to its growth rate, silk yield and fecundity. Thus, it is becoming increasingly important to turn to the new approaches of genetic engineering to produce better strains. Transferring sturdiness genes. The Indian native races of silkworm called the pure Mysore strain, though poor yielders of silk, are much more tolerant to high temperatures as well as fluctuations in temperature compared to high yielding bivalve strains. They are also capable of tolerating poor quality of mulberry leaves used as feed and appear to be more resistant to locally prevalent genes into the germline of silkworm be established. At the moment, there are two ways of doing this. However, they are yet to be perfected in the silkworm. The first one being the microinjections. Clone genes, the fibroin, are directly introduced into the early embryos by microinjecting technique. If such embryos accept the integrate the microinjected DNA into their genome, they should develop into transgenic animals harboring additional and alternative copies of the gene. Electroporation is yet another method in which electroporation and other methods of direct introduction of the DNA into dechorionated eggs may also be useful in arriving the same result. The discovery of transposons like sequences in the silkworm DNA, which are analogous to the P element of the Drosophila, raises the possibility of the exploitation of these sequences to target and inject foreign DNA. Virus mediated gene transfer. The nuclear polyhedrosis virus, which is pathogenic to silkworm, can be exploited as a vector for the introduction of the foreign genes. Experiments that use insect-derived cultured cells have already made use of this method. If you see the other applications, silkworms are generally susceptible to variety of infections like the viral, bacterial, fungal and protozoal. Methods for early diagnosis of such infections or their effective control can be developed by applications of immunological and recombinant DNA techniques. The molecular biology of non-mulberry silkworm is nearly uninvestigated. For instance, understanding the molecular mechanism of pupal diapause to avoid erotic moth emergence and reproductive biology to improve the fecundity is important in tassar silkworms. This could ultimately contribute to better silk yields. Although there are a number of races and strains of mulberry and non-mulberry silkworms employed for silk production in India. At present, there is no inventory of these. A computer-based database for the different silkworms that induces genetic and molecular characters would be useful. We'll see how silkworms are used as bioreactors. Silkworms can also be used and exploited to produce biologically important molecules other than silk fiber. Introduction of foreign genes such as alpha interferon and interleukin 3 and other growth factors into silkworm with genetically manipulated NPV genomes and expression of these products in substantial amounts in the hemolymph of larvae have already been reported. For the NPV, polyhedron is a non-essential protein for its manipulation, multiplication, but it is synthesized in substantial quantities towards the later part of the virus life cycle. Completed virus particles are embedded within a polyhedron matrix to form characteristic polyhedral bodies which serve a protective mechanism for survival of the virus, the environment. On ingestion of the polyhedral bodies by the larvae, the most common mode of infection, the polyhedron dissolves in the alkaline gut juices, releasing the viral particles, facilitating the infection. The polyhedron gene is a convenient target for genetic manipulation to introduce foreign genes for their high-level expression order to understand the action of the herbicides, pesticides and insecticides. This information should be useful in the control of harmful insects. Let us also see the biotechnology in sericulture. It is clear that the ability of the sturdy silkworm is more important as far as the quality and yield of the silk are concerned. An essential prerequisite for steadiness for silkworms is high quality mulberry leaves. Considering that nearly 90% of the silk produced in India is mulberry silk, attempts to improve mulberry plant like Morus alba itself by application of biotechnology are certainly desirable. The protein and free amino acid contents of the mulberry leaves contribute substantially to silk yield. At present, the genetics of the pure mulberry species is poorly defined, the methods of plant tissue culture and clonal propagation of high quality mulberry plants can be undertaken. Host plant improvement is even more important in the case of non-mulberry silkworms. Development of alternate food sources and synthetic or semi-synthetic diets for mulberry and non-mulberry silkworm is another area that needs attention. Let's see the status. In recent years, silkworm has become a model organism for understanding the biology of the silkworm to manipulate and reconstruct the genes to produce high quality silk. Some of the significant findings include sex-linked markers, characterization of DNA markers, construction of the early linkage maps, establishment of stable germline transformation, pharmaceutically important proteins, immune response proteins, and annotation of thousands of express sequence tags called the EST tags, construction of the bacterial and artificial chromosome libraries called the BACs, identification and characterization of the Z chromosome linked genes, demonstration of lack of dosage compensation, accomplishment of whole genome sequencing, identification of the W chromosome specific BACs, Lepidoptera specific genes, horizontal gene transfer, and characterization of the essential baculoviral genes. The development and improvement of the protocol of the silkworm transgenesis has opened new areas of the application. The nucleopolyhedrovirus is also being exploited as a vector for introduction of foreign genes. Expression of marker proteins called the luciferase and green fluorescent protein has been successfully achieved in the cell lines and larval caterpillars of the silkworm, the Bombyx mori, employing recombinant BNPV vector harboring reporter genes. Today, silkworm genetic manipulation is possible due to available of more than 400 visible mutation, 3000 diverse silkworm strains, high density molecular linkage and physical maps, complete whole genome sequence of 428 megabase page, 16,000 express sequence tags, the EST tags, and well established transgenic system. At the Center for DNA Fingerprinting and Diagnostics CDFD Hyderabad, about 400 different types of microsatellite DNA markers have been developed in the silkworm. At IIT Kharagpur and III New Delhi, under molecular biological studies on silk protein, the fibroin and sericin genes of non-mulberry silkworm, eri, muga and tussar are being carried out. Fibroin, a major protein from eri, muga and tussar, have been purified and characterized. A new and novel type of double-stranded segmented polyhedrosis virus infecting tussar silkworm have been identified at IIT Kharagpur, which will lead to developing control measures against cytoplasmic viral disease affecting tussar silkworm. Yet at CCMB Hyderabad, a prototype of web-enabled DNA database on mulberry have been developed for the first time in the world, encompassing DNA fingerprinting studies carried out on more than 150 genotypes, including 48 selected elite genotypes. At Delhi University, the transient expression of marker gene GUS in various explants from different commercially important genotypes of mulberry have been demonstrated. This success has opened up possibilities for the production of transgenic mulberry clones with desired traits in future. The Andhra Pradesh State Sericulture Research and Development Institute, Hindupur, is involved in research and development in sericulture, especially in the development of new silkworm breeds, hybrids with reference to disease resistance, including abiotic stress. Further, established centers of excellence for genetics and genomics of silk moths in collaboration with Center for DNA Fingerprinting and Diagnostics Hyderabad, India, in developing transgenic silkworm stocks. Several silkworm hybrids have been developed and authorized by Central Silk Board, Ministry of Textiles, Government of India, and some of these hybrids are under commercial exploitation. Under post-harvest technology, a process has been successfully developed for the degumming of silk with fungal protease enzyme as a valuable alternative to the existing degumming at IIT New Delhi. This will lead to improvement in the quality of fabric finished products. RNA cytoplasmic methods in India. Attempts have been made to develop molecular markers for high cocoon shell weight as well as virus resistance markers, and the characters are being established using divergent parents like Sarupat and CSR2. Sample sequence repeats called the SSRs were used to study the polymorphism between parents and for making integrated linkage maps. For the first time, 8 molecular RFLP markers linked to cocoon shell characters in silkworm have been identified by CSR and TM Mysore. That has prompted sincere utilization of the identified molecular markers for the successful application of directional breeding in the silkworm for generating improved races with high silk yield. CSRGC Hosur maintained 1053 mulberry and 438 silkworm genetic resources collected from different centers of India and abroad with main objective to establish genetic identity and racial specificity of different accessions with an attempt to identify the duplicates and to identify putative molecular markers for important economic traits. In this direction, multivoltane and bivoltane accessions characterized based on the ISSR, RAPD and EST based primers. India is also a part of International Consortium on Lepidopteran Genomics along with 7 other countries in which silkworm genomics component. Seri Biotechnology Research Labs called the SBRL labs have identified potential silkworm races, breeds specific to their more tolerance for their effective utilization in the breeding program. Analysis of genomic DNA samples of 15 selected polyvoltane silkworm races with 15 selected ISSR primers revealed that 15 silkworm races grouped into 5 and 1 isolate. Further analysis could also classify the silkworm clearly into 3 groups with susceptible, moderately tolerant and tolerant for dermal stress based on the pupation rate. Analysis also revealed that one DNA marker called the 8083000 showed highly significant correlation with thermotolerance. A network research program funded by the DBT, the Department of Biotechnology, New Delhi, on identification and mapping of DNA markers linked to NPV resistance in silkworm Bombyx mori was taken up in collaboration with major sericulture research institutes of Andhra Pradesh, Karnataka as well as CDFD, Hyderabad. When we see the prospects in this industry, when the traditional approaches of breeding program appear to be reaching natural biological limits with regard to growth rate, silk yield, fecundity, advances in gene transfer technology enhance the scope for creating the better races of the silkworm with desired characteristics. Transfer of genetic elements determining sturdiness to high producers or conversely transfer of high quality fibroin genes to sturdy races is a distinct possibility. Development of efficient and successful gene transfer system for incorporating the clone genes by germline or by direct microinjection techniques or by virus mediated gene transfer is an absolute requirement for this approach. In a parallel development, exploitation of the Bombyx mori nuclear polyhedrosis virus, based expression system for high level expression and commercial production of biological molecules of interest either through silkworm derived cell cultures or in live caterpillars has become a reality. The entire field of molecular biology of non-mulberry silkworms, especially those used for silk production in India, offers immense potential for basic research and for sericulture development, be it formulation of alternative or synthetic food sources for more efficient and domesticated rearing independent of seasonal variations and climatic conditions, or improved yield of high quality fiber to benefit the sericulturists. As a summary, we see that genomic maps constructed in the standard silkworm strains using RFLP, RAPD, ISSRs, STS and microsatellites has no doubt helped in stock identification, marker resistant selection, identification of quantitative trait loci, that is QTL characters. Further effort should be intensified to identify traits for silk production, more especially the silk fibroin, sericin, disease resistance, etc. Possibilities of production of silkworm and mulberry byproducts and more specifically their medical and biotechnological methods should be explored. Efforts to characterize useful traits of mulberry and silkworm through molecular tools should still continue with the available markers. Usage through molecular approaches collectively represent a potential gold mine of important information that can be applied to programs of genetic improvement. Molecular marker and DNA sequence analysis of extent and new silkworm germplasm collection should be prioritized for allocharacterization and understanding of a more complete genetic relationship within the breeds. Molecular markers can also be used to confirm the parentage of individuals in a breeding population for genetic fingerprinting and protection of intellectual property rights. India is yet to reap the benefits of biotechnology to bring out products in the form of biotechnology, as the same is still in infant stage in spite of making sincere but compartmentalized attempts. In view of this status, we have to consolidate our focus on developing technologies improvement of silkworm and host plants. With respect to improvement of the silkworm, we need to emphasize on the major diseases, their epidemiology for early detection of the pathogens, pathogenicity and pattern of host resistance, as well as identification of the markers for the post-cocoon traits such as denier, filament length, uniformity, tensile strength, etc. The improvement of silkworm should be based on short listing of available silkworm germplasm races for various improved economic traits through detection of respective DNA markers. Similarly, in case of improvement of host plants, the focus should include tolerance to biotic and abiotic stress as well as developing transgenic mulberry through introgression of genes for stress tolerance and silkworm disease resistance. It is hoped that recent developments in silkworm genome research provides ample opportunities to silkworm geneticists and breeders to perform such an important task, leading the country with cutting edge technologies. Here we come to the end of this session and hope you enjoyed the session. Stay with us. Thank you.